then, Joy, did you get the slide advancer? It's on the podium.
Okay, Professor Dr. Paolo Decuzzi from Genova, Italy, and he will talk about Theranostics. Thank you.
Okay. So maybe we can dim the lights a little bit. So I don't need to bore you with this slide, but just to put in the context a little bit of talk that I'm going to give in the next 15 minutes or so. So we all know that over the last 20 years, maybe 25, and if you talk to the control release society people, probably even 40 years, liposomes and many different types of nanoparticles have been designed behind the notion of the EPR effect, the non-permeability retention effect. So if you have a nanoparticle that is sufficiently smaller than these fenestrations that actually populate the tumor vasculature, these particles will passively cross the endothelial layer and get accumulated into the tumor extravascular compartment. Problem is that we know that the EPR effect, it is abundant, but it does depend on the state of the disease, on where the disease is developing. It depends on the patients, it depends on the type of disease, type of cancer. So relying exclusively on the EPR effect may be a fatal error. And there are quite a few clinical trials that right now are starting addressing this issue using liposomes, for instance, that are decorated with copper-64 or other radioisotopes, and try to really quantify and characterize the extension and also the relevance of the EPR effect. For this reason, historically, in my laboratory, we have been working on different types of nanoparticles, and we call this the armamentarium because basically we use this as different type of weapons if you want to attack the cancer. And so we have nanocubes and micelles, iron oxide nanocubes that fall within the 20 nanometers range. We have spherical polymeric nanoconstructs of pretty conventional in the 100 to 200 nanometer size, depending on what you load inside. So these are the two platforms that get into the tumor through the EPR effect, fine. Then we have larger entities, which we call discoidal polymeric nanoconstructs, which are now in the 1 micron in size, and they get into the tumor vasculature. Tumor vasculature. They are not capable of extravasating passively because they are too large, but they do deposit in the tumor vasculature, and they've been rationally designed to recognize the tortuous, low perfuse tumor vasculature. And then we have a number of other things I'm not going to talk about. So today I will just focus on what we call the DPNs, the discoidal polymeric nanoconstructs. The talk is from in silico in vitro through preclinical studies because in my lab we actually do a lot of computational modeling. We mix that with in vitro assays, which are not only conventional cytotoxicity, cell internalization assays, but also a lot of microfluidic to study the transport of nanoparticles. And we do also quite a lot of preclinical studies. So these are movies, intravital microscopy movies that we got in collaboration with a good collaborator at Boston, Massachusetts General Hospital. Andy Yoon is his name. And basically what we did is we inject into mice tail vein injection 200 nanometers polystyrene particles and 1 micron polystyrene particles. We'll look at the vasculature in the ear of the animals that was placed on the stage of a microscope, and we try to understand how these particles distribute within the vessels. Are they uniformly distributed across a cross-section of the vessels? Is there any preferential accumulation next to the vessel walls, in the middle of the vessel walls, and so on? So these movies show that 200 nanometers particles that are pretty much uniformly distributed everywhere within the cross-section. And then if you look at the 1 micron particle, they're actually rolling over the vessel walls like leukocytes would do on the venular side. And this is physiology. We have heard that for decades. So if you do a little bit of analysis, you will realize that the 2 microns particles, the 200 nanometers particle, they don't have any preferential localization within the vessel walls. The 1 micron tend to be pushed laterally by the red blood cells. And this is, again, very well known in physiology. Platelets, they work like that. and then other events. So we did develop a number of computational models that help us understanding how much this effect is regulated by the flow of the red blood cells, how much it is associated with the actual size and shape of the nanoparticles. And we realized that there is a strong component associated with actually the red blood cells. Without red blood cells, unless you are using a non-spherical particle, the spherical particles will actually follow the trajectory of the streamlines and will not marginate too much. But in the presence of red blood cells, which are more abundant, fast-moving, these are pushed laterally if they are in the 500 nanometers, 1 micron, of course larger than that, but we don't want to inject 3, 4, 5 microns particles into the human body. So there is a range in the sub-micron to micron regime where we can actually use the red blood cells to let the particles move in proximity of the vessel walls. And that's actually what we want to do if we want to target the vasculature. And then a comparison between the modeling and the in vivo experiments showing that the trends are basically there. And then we ask the question, among all different possible shapes, is there a shape that will facilitate the addition of these particles to the vessel walls? The answer is, for sure, the spherical shape, if you're looking for addition to the vasculatures, is the worst that you can get, because you have only very small numbers of receptors and ligands bonds that can be engaged simultaneously. And so discoidal particles we show computationally, we show experimentally in many different cases that discoidal particles are better than quasi-hemispherical, better than spherical, and depending on the conditions, they are surely also better than elongated, rod-shaped particles. So with this background, we move into the synthesis of a new class of nanoparticles. We call these, as I said, discoidal polymeric nanoconstructs. And we can get very different shapes. We are using discoidal, elliptical with different aspect ratios, rod-likes if you want. We can get 1 micron, 2 micron, 1.5 microns. We can even go down to 500 nanometers, but I guess you understood why we want to be in a range of 1 micron. These are somehow similar to platelets in the way they work. And one of the things that we can do with these particles, we can modulate the stiffness, which is the fourth parameter that we are playing with. The size, the shape, the surface properties, and the mechanical stiffness, the deformability. How soft are these particles? We are talking about platelets. Platelets are blood cells. They are deformable. They're not rigid. And so now we can actually work with particles that are as soft as cells, 1.5, 2 kilopascal. But we can also work with particles that are stiffer, 15 and even more kilopascal. And we can compare the behavior of these particles in terms of cell internalization. And you can see here, soft, rigid, rigid, 1 micron, 2 microns. Clearly you see the number of particles associated with these macrophages increasing. These are row cells. We have done the same with J774. We have done the same with bone marrow-derived monocytes. We're working with human monocytes as well. So the picture seems to be pretty clear. The more rigid is the particle, the easier it is for the cell to uptake it. And clearly you see here, FACS analysis, 1 micron, 2 micron, soft, rigid, rigid, rigid. The larger is the size, the stiffer is the particle, the easier is the particle uptaken by the macrophages. And when you go in vivo, this is the liver of a living animal, intravital microscopy. You see some of the particles stuck into the Kupffer cells in the liver, but most of the particles are still moving around as strains. And when you go into the tumor, the picture is completely reversed. The larger particles are sticking to the blood vessels, whereas the bluish elements, which are the red blood cells, together with some of the particles, are moving around. And if you quantify, you see that in the liver, the rigid particles accumulate much more than the soft, and the tumor, the picture is reversed. The soft, they get into the tumor more than the rigid. The soft get into the tumor at percentages of the order of 15–20% of the injected dose per gram tissue. If you remove the gram tissue and you get the absolute values, we're looking at 5, 6, 7%, which is an order of magnitude higher than what Warren Chan has documented in his Nature review papers that Joy has already presented. By the way, Warren did not consider in his analysis none of the papers that have been published on non-spherical particles. He only focused on sphericity spherical particles. And that's, to me, is a very important paper for this community because it tells us that particles are still better than drugs, but there is much more that we can do in order to improve the accumulation of these particles into the tumor. Now, this data comes from a PET analysis where the particles have been labeled with copper-64. Then we did the same thing by labeling the particles with gadolinium. And rather than using imaging, we use ICP-MS, so a completely different analytical approach, analytical chemistry now. We look for gadolinium, especially in the liver, and we compare soft and rigid. And again, the same picture that we presented through the intravital microscopy analysis emerges here. 3 times difference. And we're getting, at 48 hours post-injection, between 10, 15% of the injected dose accumulating into the liver. These are very small numbers, and they are again related to the deformability of the particles. Deformability of the particles. They are squeezing through the smaller vessels. They are avoiding the uptake by the Kupffer cells and splenic macrophages, and they are circulating long times. At 1 day post-injection, we still have about 50% of the injected dose that is still there. And of course, you can do a lot of imaging with these particles. You can do, as I said, PET imaging, optical imaging. These are data regarding the accumulation into lung breast metastases into the lungs. And you can get relaxivities that are all the way up to 60 millimolar second verse 1 with this system. Clinically, we are down here, 4 millimolar second verse 1. So it could be a very interesting blood pool agent. It doesn't extravasate. It circulates for a long time, 2 days at least. So it could be a very interesting blood pool agent. And the reason why we're getting these high values of relaxivity, we have shown it in a couple of Nature papers over the years. And I think, I don't know how much time I still have, probably nothing. I still have 5 minutes? Okay. Further questions? Yes. So I'll stop here at this point. And I just want to acknowledge the people that have been somehow contributing to this work. There are many more than this because this work was started at different institution where I am right now. Right now I'm at the Italian Institute of Technology in Genova, where we have been doing a number of the analytical chemistry analysis that I showed. And again, this is not new to the community, but it's really very nice and interesting to look at the background of all the collaborators that comes from many different disciplines. So we are looking at computational scientists, as well as chemists, physicists, vet people, computational biologists, and so on. Acknowledge the ERC, the Italian Association for Cancer Research, and fundings from the Italian government. Thank you.
Paolo,
he comes from my same institution. He cannot ask questions.
Okay. Now, I'm just wondering whether you can load any kind of drug in this discoidal, or there is a limitation because gadolinium are small atoms or molecules.
The same way we're loading the contrast agents, meaning copper, gadolinium, optical probes, is the same way that we are using for loading drugs.
Thank you, Paolo. I am a bit surprised with your soft particles not accumulating in the liver. I mean, I don't think, probably not many people are aware, I mean, I'm sure you know, that many years ago, in mid-80s, there was a very interesting paper by Harold Sherpoff and Claude Nicola in JBC that used large liposomes in terms of a micron in size, which were very deformable. And they were able to deliver those liposomes to hepatocytes by endothelial massaging. So if you have the soft
By
part,
endothelial massaging.
yes, in the liver side, because in the space of the disc, you have the erythrocytes coming and pushing the particles into the space, and then is drawn into the parenchymal region of the liver. So the same principle, I'm surprised at why it doesn't apply to your particle, because softer the particle, you should see more accumulation in the parenchyma of the liver.
Done? Okay. So clearly we haven't seen accumulation into the liver using different types of investigation technique, right? So that is a firm point. I don't know what are the difference between our DPNs and the spherical liposomes, but you should also consider that these particles are long circulating. So their deformability helps them to somehow navigate near the vessel walls, as I said, but at the same time they navigate for a long time. So platelets do not get to the space that these in the hepatocytes. So there must be something different between those liposomes and the DPNs that somehow has increased the accumulation of those liposomes into the hepatocytes. So I like to think of these particles more as platelets than your liposomes. Now, the other thing that we have to consider is, again, the shape, the discoidal shape, again, has, a very specific, allows these particles to very specifically behave into the vasculature, and that may give another reason for this difference. But it's a very interesting paper. I would like to see that. Is that available online? 1986, you said?
85, 86, 87.
Okay. Yeah. And I think that this is a good point anyways, because there are many papers that have been developed in the 80s, even before that, that belongs to the drug delivery community more than the nanomedicine community. And in many cases, we are either reinventing or getting different results, which is clearly this case. So it would be interesting to have access to that document, if you don't mind. But again, we haven't seen it. And I guess that there is a massive difference between the two platforms in that sense. And I like to think of these platforms more like platelets rather than those liposomes. Other question from people that I don't know? Yes, these are friends.
It was a great presentation. I was wondering in your simulation, because you show relatively a little larger vessels, have you looked also at capillary flow, so single file flow? How is there difference there between nano and micro size?
Yeah, I mean, the short answer is no. We haven't looked at that into that yet. We are systematically changing the size of the capillaries, but this is the place where computational mechanics and experiments, you have to find the right balance between the two. These are computations that are very intense because you have red blood cells that are occupying almost 40% of the volume of the channel or the capillary, and they are deformable. So one single case lasts about 3-4 weeks of continual running on parallel computing. Sometimes, I'm telling this all the time to my students, you have to find the right balance. It's easier to make an experiment that is going to have even higher impact or to look at computation. Computation has the great privilege, if it is working, of being predictive. And so it can guide you and gives you the trends. But, you have to find the right balance. So we can exchange emails, and as soon as I have additional data, I will send you those.
Thank you, Paolo, for a very
Thank you.
insightful
Professor
lecture.
Mughini.
And actually, thanks to Professor Decuzzi. And